So I'd like to welcome you all to today's webinar on our antibody development process. My name is Birte Ackerler, as Sarah mentioned, and I'll give you a close look at our antibody development process today, going all the way from design to product on the shelf. And I will highlight our integrated approach to design as well as testing, drawing obviously from our grand expertise on proteins, assays, antibodies over the last 20 years. So first I want to just go over the agenda real quick of today's webinar. So I'm going to give you examples on how we at Bio-Techne and R&D Systems in particular are developing new antibodies. I'll give you an inside tour with specific examples on how we address some major questions that we've been raising recently, and especially in regards to commercially available antibodies, that's becoming more and more important. So we're going to go over antigen design, why that plays a huge role to design the antigen right up front so you get the antibody of choice at the end with the application that you're interested in. Another point is host species. It's sometimes a little bit underestimated, so I'm going to say a couple of things about host species and how to choose a host species, how we do that internally. And obviously specificity testing is where I'm going to place a large emphasis. And in the end then also going into, once we have a product, how do we ensure this stays a reproducible product so that when you buy this year over year, you can rely on us developing and releasing the same quality over and over again. And in regards to reproducibility, another product line that we've introduced more recently is recombinant antibodies, and those play into that into the same topic as well. Okay. So let me start by giving you a high-level overview of the antibody development workflow, which starts by selecting a target and obviously the species of the target. At the same time, we're also defining which applications that antibody that we're interested in has to meet in the end. So that also plays a huge role in designing your antigens accordingly, and as well as some of the sensitivity specs you're after. So is it for an assay? Is it for an IHC? Those things all play a role in it. The antigen design then is really tailored towards what we're interested in as a final product. So for example, if you have an assay target, then you most likely want to start with a full-length protein, since most assays in the end will work with natural sample. So folding for the immunogen becomes important. Next is which host species, and as I said, host species selection is also a very important point. So if you have a mouse target, then you might want to choose obviously an animal that is further away from mouse. We've done this in rat. Recently, we've added rabbit. So those are two good choices. Rabbits are also described in the literature as being a better choice of animal for smaller haptens, for phosphorylated peptides. So those types of projects we're really trying to put into a rabbit rather than working with a mouse or a rat, as your failure rate will increase. And then we go through the standard process. You immunize your animal, you test. And after we choose our final clones, and I'll go through some examples on how that process is done, then we'll transfer it either to product release process, or we terminate at this point and then decide, do we want to try this again? Have we tried it and it's just not going to give us what we need, or we terminate, and that will lead to a complete termination of the project. Once we've chosen final clones, then we're going into manufacturing phase. We have QC processes in place at multiple steps. I'll give you more details on that in the coming slides. And then I'm also going to touch on product documentation, as that is an important point of us communicating to the researchers that are actually in the end using our products, to make sure that what we've done in-house is actually transferable to your labs and you can reproduce what we've seen in-house. So on the antigen design, obviously R&D Systems has spent many years, over 20 years, making recombinant proteins, making bioactive proteins. And so those types of antigens are a very good choice, usually for assay targets. Currently, it's estimated that on the market there's about 2 million antibodies commercially available from about 300 antibody vendors. However, it's estimated also that only 12 to 25% of these are actually unique antibodies made by the manufacturers. Our catalog is about 13,000 strong primary antibodies covering 5 host species, and 95% of those antibodies that you're seeing on the R&D Systems website are manufactured and developed here in Minneapolis. In addition, we're co-developing our secondaries alongside, as those are very, very important reagents as well. The applications we're covering are highlighted on the right, so we're covering everything from Luminex assays to our classic line of ELISA assays, Quantikites being one of them, Duoset being another one, as well as cell enrichment product lines and the classic antibody applications as flow, Western Blot, ICC, IHC, as well as blocking and neutralization. Okay, antigen design. The antigen design is crucial as it can drive certain applications. If your final goal, for example, is an assay, a fully folded bioactive protein is a crucial component of the whole development process. As peptides usually are more linear, presenting linear epitopes, once you're switching from your recombinant protein or your more artificial system into a final product on an ELISA assay, you will encounter that many antibodies based on peptides will not recognize the native protein and therefore will not give you what we call an assay-grade antibody. And you will lose sensitivity at best. In some cases, you will lose specificity as well, and you will just not get the final desired outcome that you're interested in. Now, the problem starting with a full-length protein, obviously, is that you have a higher possibility of cross-reactivity with other family members, so you need to be very careful when you develop these and put all the right testing and controls in place to make sure you're really only recognizing the protein of interest. Having said that, that's why a lot of people do go towards peptides. Obviously, peptides have a built-in specificity, which is one of their attractions. It is usually also used to really make sure you're detecting specific isoforms. They're fast to produce. They're not as labor-intensive as proteins. However, many peptides, if they're not properly designed, will only give you an antibody that works in a Western blot to really recognize more linear epitopes. That's been the experience across a lot of different vendors. In addition, sometimes we have to go to whole cells. This is usually one of the antigen types where we're trying to address transmembrane proteins, highly glycosylated targets. However, this is even more complex than when you get to the validation stage, as you need to make sure that you're not having cross-reactivity against any natively occurring proteins within the cell line that you're using as your background. And a lot of those will lead to high failure rates. So it's obviously not the best antigen to use, but sometimes you just don't have a choice. The last antigen that we have used in the past, and more recently we've introduced more products along those lines, are smaller compounds and haptens. Those are usually used for either hapten-specific antibodies. Many of you probably have used anti-ditch antibodies, anti-HRP. So those types of compounds and haptens, we've put a lot of emphasis in the last year. When you're working with compounds, however, since they're so small, you still need to link them to a carrier protein. And those linkages can become very, very crucial in the antigen design. So it shouldn't be underestimated. Choice of host species. The host species we're working with are mainly mouse, rat for the hybridoma process. And the hybridoma process is obviously a very, very well-established process which can give you a large number of different clones in a relatively short time frame. We've used this process for over 20 years in-house, so we have a very, very good handle and understanding of what we can or cannot expect from it. And we've generated many antibodies for both assays as well as standalone over the years. What we've recently done is that we've actually introduced a process that allows us to move a hybridoma cell line-based antibody into a recombinant antibody, and I can talk about that a little later in my talk. In addition to the classic hybridomas, we've also introduced rabbit recombinant monoclonals. Those have been described in the literature, obviously, to be a better choice for small haptens and peptides. Rabbits also don't have the complication of many isotypes like a mouse or rat. So rabbit is definitely a good animal to work with. Since we went to recombinant monoclonals for the rabbits, that will give you a high Lotto Lot consistency. The rabbit process is also a very good choice if you're after a mouse, a rat, a human cross-reactive antibody, as you want to be as far away from the original species as you can. And that's why we sometimes also on the hybridoma side, when we have a mouse target, we choose a rat or rabbit. So that's kind of the ingoing discussions we have internally, obviously, before we start any project. The last one I want to touch on is our polyclonals. Our polyclonals are mainly based on goat and sheep. So we're not going into rabbits for those, as you might recognize here. And the reason for that is that the large animals have a definite advantage in giving you a larger mass of serum for long-term supply. So these species have generated a lot of great assay antibodies for us, and as soon as we are developing an assay, we want to make sure that the quality we're producing is maintainable over years to come and is not a short-term supply. Now, rabbit will obviously give you a very limited amount of serum, so the reason then is to go to a goat or a sheep to get larger amounts of serum and maintain the same lot-to-lot consistency. All our polyclonals are antigen affinity purified to make sure that we're not starting to see drift in more non-specific reactive antibodies, and those are removed in the process, obviously affecting the yields but maintaining the quality throughout the longevity of the product line. So let me go very briefly over our Hybridoma Generation process. I'm not going to go into the details, as most of you are probably aware of how a hybridoma is generated. So we're fusing lymphocytes to myeloma cell lines, we're subcloning them to make sure we get to a monoclonal stage, and that's when we're starting to actually do testing. And the testing is really driven, again, by the specs going into the project to begin with. So we can completely tailor that ELISA testing, as well as we can add additional tests. Sometimes we add Western Blot or flow very, very early on to make sure we're not even getting to a purified material without having any chance of success. At that point, we're freezing down what we call fusion panels. Those fusion panels are usually 10 to 15 clones on average, and then going from there to a pilot scale-up and a purification. And we're repeating the application testing. So all our application testing is actually done on purified material at that point to maintain that what we develop in-house is the same thing as what we're going to manufacture in-house, which is why we've adopted the same process on both ends. Just as a side note, all those panels are available upon request. So when I say 10 to 15 clones, you will only see 1 or 2 in the catalog. However, we keep every single clone we've ever developed over the last 20 years. So those are available for testing if anybody is interested. So application testing. All the panels that we've developed are then handed off to our application testing group and validation teams. And that can include as many as 5 or 6 different groups looking at the same clone panels at the same time. So this can span, obviously, the classic applications shown in the middle, as well as our assay and array teams. And each of these will bring something different to the table. Obviously, for flow cytometry, you will use a slightly different sample type as what the IHC or ICC groups will do. So immediately, you're widening your selection of the testing materials that you're using across the same clone number, same material. In some cases, we'll have to use induction models, and I'll show you an example why those are absolutely crucial to use sometimes. We're sometimes doing comparisons with other clones, just to, again, include more wider screening and testing. And then we get back together and we look at all the data across all applications with a group of very senior scientists here in-house. And we're literally then reviewing every single clone result and making sure that the one that we put in the catalog and choose for release is the best one for multiple applications. We're always choosing clones that are meeting each application optimally, but we won't release every clone that was ever positive. So that's a little different. You will see some manufacturers that will release any clone that has any application. We've made an active decision not to approach it that way, to make sure we're really standing behind each clone we've released with the applications we're releasing them for. So what do I mean by validation across applications? This is an example where we teamed up with our Quanticline ELISA group. So we were after ApoE, and we made a gold polyclonal. Obviously, when you have gold polyclonals, that is always a little bit, it's more testing, and so you want to make sure that the sample types you're working with actually is expressing your protein of interest. So the Quanticline ELISA we had in-house, we used to actually evaluate the samples we were running on the Western ahead of time, and we quantified the amount of ApoE in the ELISA. And you see the results on the right. PBMCs are completely negative for ApoE, HepG2s have a high expression level, while the SKMEL28 cell line had a very low expression level of ApoE. When we ran the Westerns on the exact same samples, we could actually reproduce that. So that's how we did the verification of the goat polyclonal in this case. Another example I have is a phosphoantibody. This one was made against anti-EGFR phosphorylated at position 1086. And what we observed on the Western was a very nice induction on A431s treated with EGF as expected. However, when we went to the ICC, we had the untreated control, the EGF stimulated A431s, and we saw basal level detection in the A431s even without treatment. So we took a step back and we said, okay, we have a discrepancy between the Western and the ICC. It could be driven by pure sensitivity, a difference between the two methods. And so we decided to phosphatase treat the stimulated A431s to see is this basal level detection of phosphorylated EGF as is described in the literature, or is this actually detecting the non-phosphorylated EGFR to begin with? After phosphorylation, phosphatase treatment, we actually saw that all signal was abolished from the samples, and we did this for treated and untreated. And I show you the untreated sample here. So we were sure we're only detecting the phosphorylated version of EGFR in this case. As I mentioned earlier, sometimes we're developing antibodies to smaller molecules, to Haptens. So I'm just giving you one example here on how we test those. We developed anti-FITC antibodies, and we've gotten a lot of questions on, does it recognize other dyes? So what we ended up doing is we went back and we developed our direct ELISA assay. So in this case, we coated the plates with multiple antibodies conjugated to various dyes. And none of the other dye conjugates were cross-reacting with our anti-FITC antibodies. So this is one example on how we would test more compound-related, hapten-related antibodies compared to a more traditional protein target. Just to give you an example what that could look like, it is obviously completely driven by the compound target. Okay. As I mentioned earlier, sometimes we have to go to transfectant. It is not necessarily our most highly sought after testing system, but in this case, we really had very little options. So LGR5 is a stem cell marker in colonic crypts. So since it is a stem cell marker, it is actually expressed at very low levels, which makes the detection very, very difficult. So we decided to start on this work using transfectants and also bringing in a clone from the outside that had been recently released. And we started with our positive transfectants shown on the left. We also did the standard isotype controls for each. And as you can see, both our clone as well as the outside clone we brought in nicely detects a signal on the transfected in flow cytometry. So our final application wish was flow cytometry for this particular clone. And we saw a shift with both antibodies, which was a good indication. In addition, we ran the irrelevant transfectant. In this case, we used a GLUT4 expressing cell line. Again, same cell line background. We used NSO in this case. And while our clone did not recognize the NSO GLUT4 irrelevant transfectant at all, as expected, the outside clone, surprisingly enough, actually gave us a pretty high signal. So we had some cross-reactivity that we were aware of with the outside antibody. So we couldn't use that anymore as a control going forward. So we abandoned that approach. We reached out to experts in the field, and we ended up collaborating with Hans Clevers in the Netherlands on this one, who is an expert in studying this protein. So on the next slide, I show you some of the results that they actually got in their own lab in the Netherlands. And the nice thing is they did a lot of work on epitope mapping on the clones we provided them. So on the left, you see the epitopes that two of our antibodies, number 42 and number 20, are recognizing. And those regions are known to be responsible for the interaction of LGR5 with R-spondin. So they did another test. They set up activity assays based on top luciferase, and we're actually able to show that the LGR5 gets activated even in the absence of R-spondin simply using the antibody. So they provided the data back to us, and this data is now published, and we were finally able to release this clone. Since this target is very low in expression, it took us a long while to make sure that we actually saw specific data, and this finally gave us enough to feel confident about what we're releasing. Let me switch gears one more time. I wanted to spend some time on a different application that we are very invested in, which is blocking and neutralization. So I want to give you a couple examples of how we're testing that, and I want to focus on a specific topic, which is immunoregulatory receptors, which is known to be T-cell markers for the inhibition and co-stimulation. So the tests we're setting up for measuring blocking and neutralization are shown in this slide. So sometimes you cannot do an activity measurement. So if you don't have an activity assay, then we figured out, okay, how can we at least show protein-protein interaction? So we developed an assay in-house that is flow cytometry based. And the process is you have a overexpressing HEC293 transfected cell line. It's co-expressing EGFP as one of the markers to detect on flow cytometry. Then we're adding the binding partner that's described in the literature. In this case, the expressing protein was B7H7. We came back, we reacted the cells with CD28H biotinylated. This is the soluble portion of the protein. And then we can come back with strep-evident PE for detection of the interaction. If that interaction gets disrupted by any antibody, you're abolishing your signal in the PE channel, and you should hopefully see that change. Since this is a flow cytometry test, the first thing we need to do is we need to establish that flow cytometry is an application for the antibody. So we're screening all our panels. And on the next slide, I give you one example. Again, this is the B7H7 antibodies we just did in the last half year. And I show you the example for a high-expressing HEC293 transfectant on the very left. You see a very nice shift in signal upon reaction with the B7H7 antibody. You see a minor shift in K562 cell lines. Again, those cell lines are described to have a low expression for that particular target. And then as an intermediary, I'm showing you PBMC monocytes. So the antibodies we have on this particular test, this is clone number 12, performed as expected. So we knew we had the specificity of the antibody itself. So then we went on to the blocking testing. On the very left, that's the result of just adding strep-evident PE using the CD28H biotinylated soluble portion reacting that with the overexpression hex cell line, and you should see the shift that we have on the left. So that test performed as expected. In addition, we're always doing isotype controls. Again, isotype control antibodies shouldn't have any effect on the result. So the result on the isotype control also came back as expected. And then we started testing our clones, and I'm just showing you two in this case. Clone number 12, which abolished all signal in the PE channel, and clone number 34, which had no influence on the result at all. So based on this, we can show that the antibody is actually interacting with a portion of the B7H7 protein that abolishes the interaction with the CD28H soluble portion of the ligand. We then went on and wanted to turn the assay around, and rather than making more transfectin and going flow, we actually chose in this case to use Biacore. So we coated the chip with the soluble portion of the CD28H, and then we pre-incubated the two clones I showed you previously, clone number 12 shown in orange and clone number 34 shown in green, with the B7H7 soluble domain. And again, we can reproduce what we've seen in flow cytometry, so we can actually confirm the results in flow, also using a completely different method on Biacore. Okay. Sample preparation. There's been a lot of work in-house on making sure that the sample preparation is really meeting our criteria and making sure that what we send into the assays is going to give us the right result, and we're not misinterpreting any results we're getting. So why is this crucial? I'm going to go into phosphorylation-specific antibodies, as those are usually the best antibodies to highlight the problem. So we set out to make a couple lysate lots. So I'm showing you 3 different lysate lots here. The target that we chose was STAT3, phosphorylated at tyrosine 703, and we used Dowdy cell lines with interferon A2 alpha as the inducer. When we did this, we can actually, and we did the testing, not all lots are created equal. And we did this on purpose. We used cells that had been cultured slightly different. We had cells that were cultured more or less passage numbers, so you had a difference in age in the cell line. We also used different users. So we really tried to vary this as much as we can to kind of see how robust the protocols are. And what came back is we only essentially had 1 lot that would, in this case, meet our QC criteria. So we actually have a QC criteria on our lysates, and in this case, lot 1 and 2 failed. And we used this to really make sure that our protocols on the lysate preparations are very, very robust. And I'm just showing you this as an example on what you can get or expect if you are not careful on lysate preparations and you're not training people according to protocols, or the protocols aren't very detailed. The second example is HUVEC cells treated with provenidate. In this case, we used 4 different lots. And on this one, we could see very nice induction on the phosphorylation on FAK in all 4 lysates. While the first one was a little weak, it was still induced. We used those lysates to actually develop a new product. We were at that point interested in phosphorylation of eNOS. And the mouse antibody that we had tested showed very little to no really clear induction, so it depended a little bit on the lysate, and it just didn't quite mimic what we saw with the anti-FAK antibody. Now, we had 2 options at this point. We either had a very low-level detection of non-phosphorylated eNOS, that was one of the options, or the eNOS phosphorylation is just not affected by the treatment we had in place as much as the FAK. So in order to sort that out, we did a lot more experiments that I'm not going to be able to show, but in the end, we were actually able to show that the mouse anti-eNOS antibody on the very left was detecting non-phosphorylated eNOS. So at that point, we decided to drop that antibody, drop that clone, and it just went into our failed results. We redeveloped this particular target, and we finally were able to develop a recombinant rabbit monoclonal, shown on the very right, that's now released, that has the correct induction, as you can see in the picture. Again, this goes back to a point I was making earlier on, is if you need to do phosphorylated antibodies, normally a rabbit is a much better choice than a mouse or a rat. They usually respond much better to smaller haptens, and this is a clear case where we got that. So once we're done with developing an antibody, we obviously need to make sure that we maintain that quality and all that work that has gone into making an antibody over years to come. So how do we do that? This is highlighting our whole process from start to finish. Again, we're having a development phase. All applications are tested. The antibody clones get chosen based on the applications they are best used for. So the applications are established for a particular catalog product. We're going into a manufacturing phase, at which point we actually repeat all application testing. So we're not just choosing one application test, we're actually repeating all testing development had done in the beginning, that we then hand off to our release process. Once everything is packaged and bottled, we're repeating the application testing one more time before it leaves this house. So the applications are tested at minimum 2 to 3 times before any antibody hits the shelf, and before we release it to be used in your research. So that is the process for the first lot. Any lots, second lots, or higher lots, again, we make what we call a bulk lot. We make the raw material again. All application testing is repeated all over again, this time compared to the previous lot. So we need to make sure that the new lot is behaving and giving the same sensitivity as the released lot, and we're not having a drift in our product line. Again, the product gets bottled, and the testing is repeated on the bottle lot one more time before it gets released into our shipping area and meets the customer. So what do you get when you do this over years? This is an example flow antibody on granular sites, and this is our mouse CXCR2 APC secondary antibody detection, and you can see the high reproducibility between 3 lots. Another example is our caspase-1. Caspase-1 is being processed in A431s heavily, so the multiple bands are expected, and the sensitivity over 5 to 6 years, having made that product, maintains very, very reproducible across 6 lots. And we also compared this back to one of our bottled lots to make sure that the bottled lot is the same as our raw material lots. The other way of reproducing, making a very reproducible product is obviously recombinant antibodies. A lot of people have highlighted that this is one of the ways to have a high lot-to-lot consistency because you're working with a very well-defined process. You're not working with specific cell lines, you're not working with hybridomas, which on occasion can lead to drift. Every antibody company will tell you the same thing. Occasionally you will see that. All those recombinant antibodies obviously have been sequenced, so these are true monoclonals. They're all sequence confirmed before they were released. And the other beauty of this is that they allow for other manipulations in the future. So we decided to go ahead and have a very established and robust process now for making rabbit monoclonals. The reason why I bring this up is our company has chosen not to go with any cell line based. We're going from animal into PBMCs and then straight into a cloning process. And the testing is done at multiple stages during this process at small scale as well as pilot scale expressions to make sure we have no changes within scale up. The other thing we did when we established this process, we also had to co-develop an expression process for these. So the rabbit recombinant antibody expression levels or any other recombinant antibody, we're now at about 300 milligrams to 1.5 grams in a 3-liter culture. That is a large amount of antibody you can get from a single lot. again, minimizing the risk when you have lot-to-lot changes on having any changes at the customer end. Usually our traditional hybridoma methods, where you do have very high producing hybridomas, it will take about 2 times more time and effort to get to those types of levels of purified antibody in the end. And the way we actually got here was we co-developed all our vector designs. We optimized a lot of the fermentation process. We optimized our media in-house. So we really put a lot of effort into this. When we had the recombinant process established, we went back and we actually went back to our hybridomas on mouse and rat. And we've now cloned over 100 mouse and rat hybridomas, but we're not changing the isotype. So we're actually maintaining the original isotype from the hybridomas. We thought that was, we figured that was important because we wanted to make sure that we have no changes between a known hybridoma product and going into a recombinant product. We didn't want to introduce any changes on the customer end, so we actually optimized each individual isotype to get to this point. The antibodies we just released span all applications, and since we've been going from a hybridoma to a recombinant, we obviously know the full sequence, and 98% of all those hybridomas we've used are sequence-confirmed monoclonals. We're still working on more, and we now have about 40 released that are available through our standard catalog process. We've also worked with some pharma and diagnostic companies recently who were very interested in this process because they have seen drifts in hybridomas traditionally. So again, this gets out of that issue that you see occasionally. Once we're done, we need to make sure that anything we do in-house, protocol-wise, process-wise, screening-wise, is actually transferred to you as our customers, and that's where we put a lot of emphasis. So we are actually disclosing our immunogens completely, and I have an example here. In this case, it was the caspase-1 antibody that I showed you earlier. We tell you exactly which amino acids are part of the immunogen we used, as well as the accession number we used. So if we made any mistake anywhere, it would be very, very obvious here because you can check the accession number in UniProt, and you can check the amino acid locations. In addition, especially on Westerns and IHC, we've gotten a lot of feedback from customers saying, I cannot reproduce it because I don't know exactly what the protocol was. So our protocols are very, very transparent. I'll give you an example on the Western protocol. So apart from obvious things like the amount of antibody used, the cell type used, we also disclose the exact immunoblotting buffers used, and it's hyperlinked on every single one of our antibodies. We do a very similar process on the IHC ones. Again, the protocol is completely disclosed. So if you still have any questions, concerns, obviously you're more than welcome to contact us anytime. We try to give customers as much information as we can, but it's never going to be 100%. In addition to that, obviously since we've been making antibodies for over 20 years, our antibody portfolio, the data especially, is not static. So we're continuously updating the information available based on customer feedback, based on additional new research in the literature. Recently, we've added over 2,000 new data points and images to our older product lines, and we will continue to do that going forward. We can't do everything ourselves, so we also partnered up with the Affinity Binder Knockdown Initiative from Antibodipedia. So we're expecting to see more data using siRNA knockdowns, as well as other methods such as CRISPR-Cas9 coming in the next year. And we will obviously share any results that come out of that with our customers. So we hope that you will use our R&D Systems quality system to your advantage. I've touched on most of these except for the tech service. Obviously, if you do have questions, you can always contact us. Our tech service is very, very well established, and they're very skilled in what they're addressing. If they can't, they will get it straight to our development teams to answer any problems, any questions, any concerns. Okay, I just want to let you know we also have an upcoming webinar in April that will go into much more detail than I can here on our recombinant antibody technologies, and I would like to thank all of you for your time and listening.
Thank you, Berta. That was very, very insightful. Lots of information there. If anybody got quite a lot of questions already, but if you do have a specific question that you'd like to ask, we've only got time to ask Berta a selection. If you don't get your question asked right now, don't worry. We will be saving all of your questions and we'll get back to you individually. So we hope to answer all of your questions. Let me know. So we do have a question from Anders Lundqvist, who I'm just going to see if I can unmute Anders to see if he can ask his question directly to you. Okay, Anders, are you there?
Hi there.
Hi, would you like to ask your question to Berta?
Sure. I was just curious to know if you have experienced any changes in the antibody properties, for instance, in terms of affinity when you move from monoclonal production in hybridomas to recombinant expressions.
We haven't measured affinity itself, so in order for me to give you an absolute correct answer, we would have to do that. However, we haven't seen complete loss of application. So that's the data point I do have. So based on that, I would say if there's any shift, it is not leading to a drop of the complete application. So if there's minor shifts, it doesn't affect that.
Okay, fair enough. Thank you.
You're welcome.
Thank you, Anders. Actually, I have a question from looking at and watching the webinar. If, say, the application is only for something like Western Blot, what are the ways that you can determine the specificity of that antibody to make sure that you're really picking up the signal that you want to pick up?
Mm-hmm. Yeah, that's one of the open questions we get a lot of times. So if you only have a Western Blot as the only test that you're either able to test or that's the only one that actually gives you a result, is you actually want to try, you can start with the inductions on cell lines if you're after a phosphorylated target, as I've shown. That's one way of doing that. Another way is testing a wide variety of cell lines based on mRNA data that is available publicly and making sure that your Western will follow the same expression pattern. However, the difference between mRNA results and protein expression can be there, so it is only one of the options. This is one reason why we did partner up with Antibodipedia, and that will give us another data point on actual knockdown data that we would like to leverage more going forward. But that's a good question. So when you have a single application, you need to do additional testing to make sure it is specific.
Okay, thank you. We have a question about the types of validation that we do for Western blots and the application from Carol Dangelmeyer. So I'll just see if Carol's able to ask a question herself. Are you there, Carol?
Hello?
Hello, Carol. Would you like to ask Berta your question?
I would just like to know, we no longer do chemiluminescence in the laboratory. We use infrared dyes. And would the results be the same?
Let me ask you a question in return to not to get around the question. Are you using primary? You're using secondaries with IR dyes, is that correct?
Only secondaries.
Okay. Normally you should see the same result. However, IR dyes, because what you're giving up is the enhancement using an HRP system. Okay, so you're essentially giving up the enzymatic enhancement. So the sensitivity on IR detection will be lower. That's what I've seen over many targets. So as long as the expression level is still fairly robust, you should be able to reproduce the same results. The other thing that we don't have a lot of experience on is the background on IR dyes on membranes, but I'm assuming that that's all corrected for. So the only point of concern would be the sensitivity levels. They will change going from a chemiluminescent system to an IR.
I see. We've also had some problems with non-specific phosphoproteins, phosphoantibodies to our proteins. It appears they also pick up non-phosphorylated protein. Do you do any kind of specific testing to ensure that the regular protein doesn't get picked up as well?
Yeah, so when we develop phosphoantibodies, it takes us a long while because of all the controls we run. So let's go back to the example I had on the eNOS, where we did see some induction, but we had a high basal level. So exactly. You have, yeah, exactly. So you have multiple options at that point. So what we ended up doing is the first thing is you can tighten down your primary. If you have a very high affinity antibody, you will overwhelm the system, and you can force any phosphoantibody pretty much to recognize also non-phos if you really intend to do so. We've done that test 5 times, 6 times over, and you can get that. So that's why it's crucial that you actually get the antibody titer into the correct range to make sure you're only recognizing phosphorylation. The other thing is you need to do the dephosphorylation in a lot of instances to show that your basal level detection without induction is the phosphorylated protein to begin with. If that doesn't change upon phosphorylation, then I would abandon that particular antibody personally.
Okay. Thank you.
You're welcome.
Thanks very much, Carol. Great question. We have a question from Corrine Ennisser. I hope I'm pronouncing your name right. My apologies if I'm not. I'm just going to see if I can unmute you, Corrine. Okay. Corrine, go ahead. Are you there?
Yes, I am there. Thank you very much. Thank you for the presentation. I really enjoyed it. So I was wondering, what is your experience with immunohistochemistry success regarding transmembrane proteins, whether you went with a peptide approach or recombinant proteins?
Okay, so your question, let me just clarify this, is if we've seen the same success rate with peptides versus full-length proteins. Is that correct?
Yeah. Yes, that's correct. Yeah.
The peptide development process for IHC antibodies has been improved over years, not just by us. It's been approached by a lot of different companies. And yes, you can get the same success rate.
And so I guess it would depend on your target. So if it is a target that is like a transmembrane protein, would that change in terms of success rate? Sorry for the background noise.
On transmembrane proteins, I would highly go towards the extracellular domain rather than just a peptide, just to increase your chances. When you go to peptides only, the concern I would have is because on IHC you have another level of complication. It's not a soluble method at all. It's cross-linking everything very heavily in the tissue. So going to an extracellular domain, if you can express that, purify that to at least 80% or higher, that will give you a higher chance of success in IHC.
Thank you very much.
So.
Thank you very much, Corrine. We have a time for a couple more questions. A good question here from Jacinto Villanueva. So let me just see if I can unmute you, Jacinto. Are you there, Jacinto?
Yes, I am.
Hi, Haas.
Good day. A very nice webinar. I really appreciate this. Thank you.
Thank you very much.
So my question is, now that you guys have gotten into recombinant antibodies, have you explored labeling of your primary antibodies directly for detection?
Yes, we have.
Is that going to be an April seminar?
Yeah, it's that is correct. Sorry, it's not that I'm skirting your question, but that is really a lot. There's a lot of details and a lot of work we've done on the recombinant platforms with essentially 4 different species at this point. So it would kind of go outside, but yes, that was one of the things we've investigated in, and among us, like tagging antibodies, all that manipulation that really opens up once you have a recombinant clone in hand.
Oh, that's excellent. Thank you.
So I hope you're going to join us in April.
I will. I'll do my very best. Thanks.
Thank you.
Thanks very much. Okay, a few more. So we have a question. I'll ask this one directly from Nicholas Bartholomew. His question is, do you see any differences in the antibodies that you choose for those used for ICC applications and IHC applications?
Yes, we do. So in some instances, and we've seen this very repeatedly, is sometimes you will not be able to find a clone that will work in both ICC and IHC. I've talked to some researchers that kind of treat that application as the same. However, I would just like to put in one word of caution that IHC does require a very harsh antigen retrieval because you're cross-linking everything to begin with. So I would treat them as 2 separate applications, and so that's what we see when we actually screen. Sometimes we see antibodies work beautifully in ICC, and they will not either recognize anything in IHC, or they will give you a very high background. So it's really not necessarily predictable.
Okay. And do you see, is there any predictability between the antibodies when you're validating for a number of different, are there any applications, are there any predictors of what might be good for an antibody perhaps in a direct immunoassay opposed to one for a Western blot or flow cytometry per se?
That's a tricky question to answer. I have not seen any true indicators for predicting an antibody from the start whether or not it's going to work in a specific application. There is a tendency for peptide antibodies to work more in Western blots versus any application that requires a fully folded epitope. You will see overlaps in clones between assays and flow cytometry, as well as more of a neutralization. So any application that requires a folded protein, those have a tendency to work on the same clones versus a Western type of application. So some of our best Western antibodies aren't necessarily the best assay targets, and reverse, even within the same panel. So they've all seen the same exact immunogen, and even with that, you will see that split a lot of times. Okay,
thank you. I think we'll wrap up there. There are a few more questions that we've been asked, but what we will do is, as we don't quite have enough time to answer them all now, we will email, we're collecting all of these questions, and we will email you directly the answers to the questions that you do have. So I'd like to thank everybody for attending this webinar today, and to Bert Agler for her time and presenting a really insightful webinar on all of the work that can be done into antibody validating and reproducibility. We will be emailing in a day or so the link to this webinar where you'll be able to find it and watch it again if you so wish at your own leisure. So thank you very much.